In this video, we are going to discuss about DNA and RNA purification. Now let's start with DNA purification. Now, in the process of the DNA isolation and purification, there is disruption and lysis of the cells. After the disruption and lysis of the cells, there is removal of proteins and other contaminants. And at last there is recovery of DNA. Okay, so this DNA purification can be, broadly classified into these three stages. That is, you disrupt and lyse the cell. After that, you remove the protein and other contaminants. And at last, you do the recovery of the DNA. Now, the cell are lysed with the help of a detergent. Okay, detergent is used to break down a cell, and then cellular debris is removed by centrifugation. And after doing this, the remaining soluble material is mixed with phenol or 1:1 mixture of phenol and chloroform. Okay, you lyse the cell with the detergent, then you remove the cellular debris with centrifugation, and whatever is left after this remaining soluble material, that remaining soluble material is mixed with phenol or 1:1 mixture of phenol and chloroform. Now this organic solvents, that is phenol or phenol chloroform, they precipitate proteins and they leave the nucleic acid in the aqueous solution. Okay, so you lyse the cell, you then remove the cellular debris by centrifugation, and whatever remaining substance is there, you mix it with phenol and chloroform, and due to this step, the proteins in that mixture gets precipitated and a aqueous solution is formed which contains nucleic acid. Okay, so after you mix that remaining thing with the phenol and chloroform, the proteins get precipitated and the nucleic acid gets there in the aqueous solution. So now your focus will be to remove this nucleic acid from this aqueous solution. Okay, so after that, from the aqueous solution, the nucleic acid is removed. Now, if the protein content in the cell extract is very high, then we use proteases such as pronase and proteinase K to cleave the proteins before phenol extraction. Okay, we do cell lysis, we get cell extract. Then that cell extract, cell debris is removed by centrifugation, and whatever the remaining substance is there, we apply it the phenol extraction thing. But if in that thing the protein content is very high, then before phenol extraction, you apply protease enzyme to it, such as pronase and proteinase K, so that the proteins will be cleaved, and then you apply it to the phenol extraction so that proteins get precipitated and you get the nucleic acid in the aqueous solution. Okay, so you remove proteins and you want to isolate DNA, but RNA can be there in the aqueous solution, right? So this RNA is removed from the preparation by the treatment with RNAs. Okay, so the RNAs will digest RNA and you will be remained with DNA only. So that is how you isolate DNA. So this is how the process looks like. You have cell extract. Okay, this you get after the cell lysis. Okay, after the detergent, after putting the detergent lyse the cell and you get a cell extract. Okay, this thing. Now this thing is subjected to centrifugation. Okay, now after centrifugation you have cell debris, and in this solution you have DNA, RNA and protein. Okay, after that you mix a phenol in it, okay, phenol chloroform solution in it, and then after putting the phenol chloroform solution in this thing, you centrifuge it, and after centrifugation you get a aqueous layer. That aqueous layer has DNA, RNA, and then you get coagulated proteins and then a phenol layer as well. Okay, so you after this you will isolate this aqueous layer and you will put RNAs, and then you will get in last pure DNA. Okay, so this is how the removal of proteins by organic extraction and enzyme digestion looks like. Okay, so you take cells, you put detergent in it, then that detergents will cause the lysis of the cell, you will get cell extract. Okay, now in this cell extract you will apply centrifugation. Okay, to this cell extract you will apply centrifugation. After centrifugation you will get cell debris and this solution. You will remove this solution and put it in the another tube. Okay, now in another tube you will also put phenol chloroform, and then you will subject this to centrifugation. Now after centrifugation you will get a phenol phase, then the aqueous phase, and in between there will be a layer of coagulated proteins. Now these two things are of no use. You isolate this aqueous layer and then you isolate DNA from that. Okay, that aqueous layer now has DNA and RNA. So after purification, after getting that aqueous solution, you can concentrate the nucleic acid present in the aqueous solution using ethanol. Okay, nucleic acid of the aqueous solution can be concentrated by using ethanol, and this is known as ethanol precipitation. Now, ethanol precipitation involves the precipitation of DNA using absolute ethanol with monovalent cations. Okay, you subject the aqueous solution of DNA with absolute ethanol and with monovalent cations, and you keep it at the temperature of -20 degrees Celsius or less. Okay, so by doing this you will then isolate your DNA from the aqueous solution that you got after doing phenol extraction, and this whole technique is known as ethanol precipitation. Plasmid purification. Now in plasmid purification what is done is the bacterial cell extract is deproteinized, then RNA is removed, and then the DNA is concentrated by ethanol precipitation. Okay, so this is what we do in plasmid purification. Now let's look at in the details. Now in plasmid purification, it is necessary to separate the plasmid DNA from the large amount of bacterial chromosomal DNA. Okay, so there are proteins, there are RNA, there are other all impurities, and the challenge here is that you have to separate your plasmid DNA from the bacterial chromosomal DNA as well. So that's why it becomes quite sensitive, I will say. Now the separation of plasmid DNA from the chromosomal DNA depends upon the physical differences in the conformation and size of the plasmid DNA and chromosomal DNA. Okay, there is a physical difference between the plasmid DNA and chromosomal DNA. There is a difference in the conformation of both of these. There is a difference between the size of both of these, and on the basis of that difference, those physical differences, we separate the plasmid DNA from the chromosomal DNA. Now as you know, the plasmid DNA is circular and the bacterial chromosomal DNA is also circular. But while the preparation of cell extract, the chromosome is broken down into linear fragments. So in the cell extract, you have a circular DNA, that is your plasmid, and then you have a linear DNA, that is your chromosome. Now you have to just figure out how to separate the circular ones from the linear ones. Okay, so the first step you can say, you break down the chromosomal DNA, the circular chromosomal DNA, into linear fragments while preparing the cell extract. Now plasmids are the covalently closed circular DNA which are present in the supercoiled conformation. Okay, plasmid shows supercoiled conformation. Now if one polynucleotide strand is broken, then the double helix reverts to its normal relaxed state, and then the supercoiled molecules can be separated from the non-supercoiled DNA. Okay, so the thing is that the DNA, the chromosomal DNA, is in the non-supercoiled form. The plasmid DNA is in the supercoiled form, and then this supercoiled molecules are separated, and that is how you separate the plasmid. So one method of separation of the supercoiled molecules from the non-supercoiled DNA is through alkaline denaturation. Okay, by doing alkaline denaturation, you can separate the supercoiled molecules from the non-supercoiled DNA. Now, if you add sodium hydroxide in the cell extract which contains linear DNA and supercoiled DNA, then the hydrogen bonding in the linear DNA molecule, that is non-supercoiled DNA, is broken and the double helix unwinds, and then the two polynucleotide chains can be separated. Okay, so the thing is that in your cell extract you have linear DNA and you have supercoiled DNA. That supercoiled DNA is your plasmid DNA. The non-supercoiled DNA or the linear DNA is your bacterial DNA, and you have to separate the bacterial DNA from the supercoiled DNA, that is the bacterial DNA from the plasmid DNA. So when you add this sodium hydroxide now, the hydrogen bonding of non-supercoiled DNA is broken. Okay, it does not affect the supercoiled DNA, but the non-supercoiled DNA's hydrogen bonding is broken, and that is the reason why the double helix of this non-supercoiled DNA unwinds, and that is how you separate the two polynucleotide chains. So in this separated two polynucleotide chains, you can add acid, and after you add acid, the denatured bacterial DNA strands that just formed because of the addition of the sodium hydroxide, the denatured bacterial DNA strands reaggregate into a tangled mass. And then this insoluble network of this tangled mass of this bacterial DNA strand is pelleted out by centrifugation, and then you have your pure plasmid DNA in the supernatant. Okay, so let's go again. So you have your cell extract have linear DNA and your plasmid supercoiled DNA. You add sodium hydroxide to it. Now this sodium hydroxide breaks the hydrogen bond of the bacterial DNA molecule, which is linear and non-supercoiled. Now because of this, the double helix unwinds and the two polynucleotide chains, that is the two strands of the bacterial DNA, get separated. After that, after the separation of these two strands of the bacterial DNA, you add acid to that solution. Now this acid, the addition of acid causes reaggregation of this denatured bacterial DNA strand, and there is formation of tangled mass. Now this tangled mass, this insoluble network, is pelleted by centrifugation, and when you subject centrifugation, then the pellet will contain this denatured bacterial DNA strand which is insoluble. Okay, that would be the pellet, and in the supernatant you will be having pure plasmid DNA. So that is how you pure the plasmid DNA, by using sodium hydroxide and an acid. Now another method for plasmid purification is Cscl buoyant density gradient centrifugation, and this process we do in the presence of DNA intercalating dye known as etbr, that is ethidium bromide. Okay, so this is your just the another method for the plasmid purification, that is Cscl buoyant density gradient centrifugation in the presence of etbr. Now etbr intercalation causes the local unwinding of the DNA helix. Okay, if the etbr intercalates in the DNA helix, then that DNA helix will be unwind, and if there is unwinding of the DNA helix, then that reduces the molecular density of the DNA. Okay, now the thing is that the large linear non-supercoiled genomic DNA bind to more etbr. Okay, to your bacterial DNA more etbr binds, and to supercoiled DNA less etbr binds. Okay, you put etbr in the solution. In solution you have both plasmid DNA and bacterial DNA, but etbr will bind more to the bacterial DNA and etbr will bind less to the plasmid DNA, and because of this there will be difference in the topology. That is, there will be difference in their molecular density. Okay, after the addition of etbr, the molecular density of the genomic DNA or the bacterial DNA and the molecular density of your plasmid DNA will be having a difference, and that difference in the topology, we will use that difference to separate the supercoiled plasmid DNA from the large linear genomic bacterial chromosomal DNA. So etbr is playing major role. Okay, major role. etbr binds, density reduces. etbr binds more to the bacterial DNA, that is why more density reduces. etbr bind less to the supercoiled DNA, so there is no much difference in it. So, and this causes the difference in the densities of your bacterial DNA and supercoiled DNA, and on the basis of that difference we are going to separate them. So consider, so this is how it looks like. You took your aqueous solution in which there is your linear DNA, that is bacterial DNA, and then there is your plasmid DNA. Okay, now you put you subjected etbr. Okay, you put etbr into it. Now etbr, what will it do? It will reduce the density of this bacterial DNA more than this supercoiled DNA, that is plasmid DNA, and then you will subject this thing to centrifugation. Okay, so after centrifugation, obviously the supercoiled DNA will be at the lowest position because it has more density than your linear double-stranded DNA or nicked circular DNA. So you can remove this part and then you can easily isolate your supercoiled DNA, and that's the whole point of doing this experiment. So separation of this supercoiled DNA from non-supercoiled DNA is done on the basis of density gradient centrifugation in the presence of ethidium bromide etbr. Okay, so ethidium bromide binds the DNA molecule, it intercalates its adjacent base pairs and causes the partial unwinding of the double helix. Now this unwinding of the double helix decreases its buoyant density. Now the thing is that this supercoiled DNA has no free ends, and that is the reason why it has very little freedom to unwind, and that is the reason why to the supercoiled DNA less amount of etbr intercalates as compared to linear DNA or nicked circular DNA. Okay, so that is the reason why the decrease in the buoyant density of the supercoiled DNA, that is your plasmid DNA, is very much less. And after centrifugation, that is the reason why this supercoiled molecules, this supercoiled plasmid molecules forms a band in the etbr Cscl gradient at a different position than the linear DNA or the nicked circular DNA. We saw in the diagram, right? It was at the lowest position. Okay, so this supercoiled plasmid DNA cannot be easily unwound, and that is the reason why much less etbr intercalates in it. But the linear DNA molecules are not topologically constrained, and that is the reason why they absorb more etbr molecules, and that is the reason why this causes an overall decrease in their molecular density. So yeah, that's all for this Cscl gradient. Okay, Cscl beyond density gradient centrifugation in the presence of interconnecting dye etbr. etbr will reduce the density of your chromosomal DNA. It will not reduce the density of the plasmid DNA. After that, you will do centrifugation, and as there is different in the density, you will get different bands of the chromosomal DNA and a different band of the plasmid DNA, and from that band of your plasmid DNA you can then isolate your plasmid DNA, and that is how the purification of plasmid DNA is done.